Hello, my name is Brian McDonald, and today I will be discussing direct-to-consumer personal genomics. Today we will go over what a genome is, how it is read, and what it maps out. Then we will look at one of the companies providing direct-to-consumer genomic testing, FamilyTreeDNA.com, some of the pros and cons of the commercialization of Ancestry, and finally I will discuss my research process, what I learned, and what I still hope to learn. Per our Concepts of Biology textbook, genomes are mapped by locating genes on the chromosomes. Genes are a sequence of nucleotides that, depending on their locations on chromosomes, will code for different proteins. We will discuss this in better detail in the next slide. These locations and types of genes are mapped out, creating an individual's genome. If we map at least two individuals' gene sequences, then we can compare those two genomes to determine the rate of similarity. A more practical application of genome mapping is comparing similarities and differences to single out correlations for genetic diseases. So where does the genome map lead? DNA sequences lead to the transcription of RNA. RNA is very similar to DNA, except that the nucleotide thymine is swapped for uracil in RNA. The subsequent sequence of RNA is read by your cells to denote what amino acids and ultimately proteins will be grafted. These proteins are then put together to create you. FamilyTreeDNA.com is one of the companies currently providing direct-to-consumer human genome testing and mapping. They collect samples from customers and compare those to known genome maps. The customer uses supplied swabs to sample the inside of their cheeks and mails that to FamilyTreeDNA. Once the company receives the sample, they send it to their lab, where half of the provided samples are stored for contingency and the other half is tested. The lab separates the DNA from the rest of the cell components using an enzyme called proteinase K. Family Tree DNA's lab then uses polymerase chain reaction to create copies of the DNA sample. These copies are then either stored or analyzed, where the sequence of genes are read and compared to other known genomes to produce possible ancestral relationships. What are some pros and cons of the direct-to-consumer model? A study of personal genomic data performed in 2014 found that the direct-to-consumer model allowed for a large and ever-increasing sample size of genomes across the planet. A benefit of this data is more accurate genetic disease tracking. One issue, however, with the direct-to-consumer model is that people may place too much trust in the system and jump to their own conclusions. One study in 2017 found that some customers changed how they took their medication without consulting a doctor after the results of genome testing. What did I learn? This project forced me to dig deeper into the textbook and other sources. I learned more about genome sequencing, the difference between DNA and RNA, and genes and codons. I had heard of guanine, cytosine, adenine, thymine, and uracil before, but I did not know that thymine and uracil were analogs in DNA and RNA. I better understand genome mapping and ancestry websites now, but this is an introductory course, and I am sure there is much more to learn. A question I have to further research is, since each genome is unique due to crossovers during meiosis and the random nature of which gamete someone can get from their parents, how similar does a genome need to be before it is considered an ancestor in the colloquial sense? Regarding AI use, I did not intentionally use any artificial intelligence or large language models for this project. AI is pretty pervasive in search engines, so there might be something in there, but I try to dig deeper and ensure I don't rely on AI. In conclusion, we went over how DNA is read to create genome maps, and we went over why that is important, because it makes us who we are. Then we went over one of the direct-to-consumer personal genomics companies, FamilyTreeDNA.com, and some of the pros and cons of the DTC model. Finally, I talked about my experience with this class and project, and a little of what I still have questions about. This slide is my references throughout the presentation. I looked at two journal articles about personal genomic data, the Family Tree DNA website, and the Concepts of Biology textbook.